Good morning students, good morning friends. Today we will discuss about translation in eukaryote, that is protein biosynthesis proper, and we will discuss this topic in comparison with prokaryotic translation. So first we will discuss what do we mean by translation, that is definition. Then we will discuss about the requirement. Then we will discuss the various steps in the translation process, that is uh, activation of tRNA, then initiation, elongation, and termination. In initiation, we will discuss about the first step is mRNA activation, then formation of ternary complex, ribosomal dissociation, formation of 43S initiation complex, then formation of 48S initiation complex, and formation of 80S initiation complex. This we will discuss in initiation. In elongation, there are three steps. First one is the attachment of amino acid tRNA to the A site. tRNA attachment to the A site. Then peptide bond formation. And third one is the translocation of ribosome. In termination process, we will discuss about the stop codons and release factors. Release factor 1, 2, 3. Coming to the definition, all of you know the definition of translation. The information which is present in the DNA, it is transcribed into the mRNA in the form of codons. Codons are nothing but the triplet base sequence which is present in 5' to 3' direction, and that code for particular amino acid, that is known as translation. In translation, the information which is present on the mRNA in the form of codons get translated in the form of amino acid. In definition, this one is the mRNA, this one is the 5' end, these are the codons, this one is the 3' end, this is the first, second, third, fourth, fifth, sixth, seventh. Each codon code for particular amino acid, and in translation process, in translation they will give different amino acid: 1, 2, 3, 4, 5, 6, and 7. So this one is the amino acid 1, this is 2, this is 3, this is 4, this is 5, this is 6, and this is 7. Each codon represent amino acid. There is peptide bond, and peptide will synthesize in the direction N terminal to carboxyl terminal. So this is mRNA. The information which is present in the form of triplet, it gives particular amino acid, and finally we will get the peptide or protein, and this is known as translation. Now coming to the requirement, all three types of RNAs are required in the translation process, that is mRNA, rRNA, and tRNA. We require the ribosome, these are the machineries. Machineries for the protein synthesis. Ribosome, they are composed of rRNA, which has catalytic role, and proteins. Then various factors are required for the translation process. They are known as translation factor. For the initiation, we required eukaryotic initiation factor. For elongation, we required eukaryotic elongation factor. And for the termination, we required release factor 1, 2, and 3. Now we will discuss about the various steps in the translation process in eukaryote with reference to the prokaryotic translation. Now, first step in the translation process is the activation or charging of tRNA. First step in the activation of tRNA is the activation of amino acid. This amino acid, with the help of ATP in presence of enzyme, that is amino acyl tRNA synthetase. It is formation of this enzyme AMP and amino acid complex. This is activated amino acid. Now this active amino acid, it react with tRNA molecule, or it get attached to the tRNA molecule, and there is formation of charged tRNA or activated tRNA. For example, this one is the tRNA. Here is the attachment of amino acid. This one is the CCA end. It can be written as tRNA CCA end, and this is the OH of this A, and to this OH there is attachment of amino acid, and this is known as activated tRNA or charged tRNA, and this is the first step in the translation process. Then we will move to the second step, that is initiation of translation. In the initiation of translation, the first step in eukaryote is mRNA activation. This step is not in prokaryotic translation. In mRNA activation, all of you know that the structure of mRNA. This one is the mRNA, this one is the 3' end, this one is the 5' end. There is starting codon A, U, G. There are the stop codons. This is the mRNA. In mRNA activation, what happened with the help of different factors? For example, 4F, it is formed with the help of 4G plus 4A plus 4E. This factor along with ATP help in the activation of mRNA. This one is the cap binding factor. This factor is known as cap binding factor, and this 4E helps in the recognition of the cap. Recognition of cap of mRNA. At the 5' end there is there is methylguanosine triphosphate cap in the mRNA, and this 4E help in the recognition of the cap of the mRNA. This 4A, it removes the secondary structure. Removes the secondary structure of mRNA. Due to the coiling, there is secondary structure in the mRNA which is removed by the 4A. 4G is the part of 4F, and 4F is the cap binding protein in the mRNA. This 4F addresses the molecule to the ribosomal unit, and this results in the formation of, this one is the 5' end, and at the 5' end there is 7-methyl guanosine triphosphate cap, and to this cap there is attachment of the 4F. In the next step there is attachment of the 4B plus poly A binding protein. This one is the 4F. There is attachment of the 4B. This one is the AUG. This one is the 3' end, and there is attachment of the poly A binding protein, and there are multiple A at the 3' end of the mRNA. So what is the role of 4B? 4B help in the recognition of AUG sequence, and poly A binding protein along with this poly A at the tail of the mRNA, they help in recruitment of 40S ribosomal unit to the mRNA. So these are the different function of these factors, and this is activated mRNA, and now it is ready to bind with 40S ribosomal unit. There is another marker sequence upstream to the AUG. This marker sequence upstream to the AUG, which is complementary to the 18S ribosomal complex. This one is the 18S ribosomal complex or ribosomal subunit. This marker sequence, which is present upstream of the AUG, and it is complementary to the 80S subunit of ribosomal RNA. It is known as Kozak consensus sequence. While this type of sequence in prokaryote, it is known as Shine-Dalgarno sequence. In prokaryote, it is known as Shine-Dalgarno sequence, which is marker sequence present upstream of the AUG, and it helps in the recognition of this initiation codon AUG. So this is the first step, mRNA activation. Now we will move to the second step. It is ribosomal dissociation. In this ribosomal dissociation, this 80S subunit, it is dissociate into 40S and 60S unit in eukaryote. This 40S unit in eukaryote, it is composed of 18S rRNA and near about 34 proteins. While the 60S, it is composed of 5S rRNA, 5.8S rRNA and 28S rRNA along with near about 50 proteins. So for the ribosomal dissociation, the factor required is, that is initiation, eukaryotic initiation factor 6. It is required for the ribosomal dissociation. This is the second step. Then third, there is formation of ternary complex. Ternary complex is methionine tRNA plus eukaryotic initiation factor 2 plus GTP. This one is the GTP, this one is the factor 2, and this one is the methionine tRNA, and these three, it is known as the ternary complex, and this is the third step in the initiation. After the third step, first is the mRNA activation, second one is the ribosomal dissociation. In this ribosomal dissociation, this ATS, this group 40S and 60S unit with the help of this factor that is known as eukaryotic initiation factor 6. This one is the 40S unit, and it is stabilized by the factor that is eukaryotic initiation factor 1 and eukaryotic initiation factor 3. To this 40S unit, there is attachment of this ternary complex there is attachment of this factor 2 along with GTP, and there is attachment of this ternary complex. This is methionine tRNA plus eukaryotic initiation factor 2 plus GTP. They get attached to this 40S ribosomal unit to form 43S pre-initiation complex. This is known as 43S pre-initiation complex. To this 43S pre-initiation complex, there is attachment of mRNA. This one is the mRNA, 5' end, 3' end. This one is the AUG, and to this AUG there is attachment of this methionine tRNA, and this complex is known as 48S initiation complex. So first in the initiation is mRNA activation, followed by ribosomal dissociation, followed by formation of ternary complex. Then there is formation of 43S pre-initiation complex after the attachment of this ternary complex to the ribosomal unit, and then mRNA attached to that unit, and there is formation of 48S initiation complex. To this 48S initiation complex, there is attachment of this 60S unit, and it results in the formation of this active ATS complex, initiation complex or active ATS unit. It is composed of mRNA, then methionine tRNA. This is difference, that is in prokaryote it is formyl methionine tRNA, in eukaryote it is methionine tRNA. This one is the AUG codon. This is the 5' end of mRNA, this is 3' end, and this required the factor that is eukaryotic initiation factor 5, and after attachment of this factor 5, what is the role of this factor 5? After attachment of this factor 5, all other factor will dissociate, and it also helps in the association of 60S and 40S ribosomal unit to form a ATS ribosomal unit. So here is the formation of ATS initiation complex. This ribosome, it has 3 sites. This one is the peptidyl site, this one is the acceptor site, and there is exit site in the larger unit only. So this methionine tRNA occupies the P site, peptidyl site. The incoming tRNA that will occupy the A site, and deacylated tRNA will exit from this exit site. So this is all about initiation of translation. We discuss here the eukaryotic elongation factor 6, which is responsible for dissociation. Then factor 1 and 3 It prevent the association of these 40s and 60s, means stabilize this 40s ribosomal unit. Then there is attachment of this ternary complex, which is activated by eukaryotic initiation factor 2. Then eukaryotic initiation factor 5. After this attachment, all other factors will release, and it is responsible for association of 40s and 60s unit. Now we will discuss about the elongation process. Elongation will be described in three steps. First is the attachment of tRNA, activated tRNA to the A site. Second, peptide bond formation. And third one is the translocation of ribosome. And for this, two important factors are required, that is eukaryotic elongation factor 1 and eukaryotic elongation factor 2. The energy for this attachment of tRNA to the A site, it is done by the eukaryotic elongation factor 1 plus GTP, and translocation of ribosome, the eukaryotic elongation factor 2 is required along with GTP. So this is ATS ribosomal unit or ATS initiation complex. To this ATS initiation complex, this one is the incoming tRNA with second amino acid. This it is done with the help of factor 2, eukaryotic elongation factor 1 plus GTP, and this one is the second amino acid. This one is the second. This one is the tRNA, activated tRNA. The first step is attachment of this activated tRNA to the A site. This will get attached to this site. AA2. These are the anticodons on the tRNA. You recognize the codon on the mRNA. Now the second step, the peptide bond formation. This now methionine tRNA, this methionine from this tRNA will move to this, and there is peptide bond formation between these two amino acids. And this is with the help of peptidyl transferase activity, which is present on the larger subunit of ribosome. Then there is again and the third step is the translocation. Move one codon away, and this one is the ribosome, this one is the smaller unit, this is mRNA, this is A site, this is P site, and this is exit site. Now there is attachment of the the third step is the translocation of ribosome. Ribosome will move one codon. So this is the second tRNA. This is methionine. This is second amino acid, and deacylated tRNA will be released from the exit site. This one is the exit site. This one is the P site. This is the acceptor site, and acceptor site will now free for the attachment of tRNA to the A site. So this will repeat and repeat again and again, and to complementary by the recognition of codon which are present on the mRNA, there is elongation of this peptide will occur. This is methionine, this is second amino acid, this is third, this is fourth, this is fifth, this is sixth, and so on. The peptide will form with recognition of codon on the mRNA. This is all about elongation, and these are the two factors which are required for the elongation. Now we'll discuss about the termination process. In In termination, the two things are important. First one is the stop codon, and second one is the release factor. So these are the three stop codon, that is UAA, UGA, and UAG. When this machinery encounter this stop codon, there is attachment of release factor which has structurally resembles to that of the tRNA. Means this is mRNA, this one is stop codon UAA. When they encounter stop codon, there is attachment of release factor which is structurally similar to that of the tRNA molecule. This one is the release factor 1 and this one is the release factor 2. These factors get attached to the stop codon and they bring about the hydrolysis. This one is the 6th amino acid and this one is the tRNA end. So this bring about the hydrolysis of this bond, and after the hydrolysis this peptide will release from this complex. So after the attachment of release factor, there is release of peptide chain, then the ribosomal unit, that is 60S and 40S ribosomal unit will release, then tRNA will release, and various release factors and all other assembly will release, dissociate from the complex. So this is the role of release factor and this is the termination. So in today's topic, today's lecture, we discussed the activation of tRNA, then initiation, elongation, and termination. In initiation, we discussed the mRNA activation, then ribosomal dissociation, formation of ternary complex, and formation of ATS unit. In elongation, there are 3 steps: attachment of activated tRNA to the A site, peptide bond formation, and translocation. And in termination, we discussed the stop codons and release factor. The release factor has structurally similar to that of the tRNA. It get attached to the stop codon and stop the protein synthesis, and now peptide will be available. This will then go for the post translational modification, and there is protein targeting. This is all about protein translation. We will discuss some points on the PowerPoint presentation. Now we will discuss translation in eukaryote in this PowerPoint presentation. Thank you, Dr. Trupti Ram Teke, for preparation of this PowerPoint presentation and animation used in this PowerPoint presentation. We will discuss about translation in eukaryote, difference between prokaryotic and eukaryotic translation, regulation of translation in short, and inhibitors of protein synthesis. These inhibitors of protein synthesis, it is particularly useful in NEET preparation. These are the headings under which protein synthesis will be described. These are the different factors which are required for initiation, elongation, and termination of translation process. These are the functions of various factors in the translation process. For example, eukaryotic initiation factor 2, it is required for the formation of ternary complex 1 and 3. It binds with the 40S unit and prevent the reassociation of 40S and 60S unit. Then 4B, it binds to the mRNA and helps in the recognition of AUG sequence. 4A removes the secondary structure in the mRNA. 4E is responsible for recognition of the gap. 4G is the part of 4F complex, and 4F address the mRNA to the ribosomal unit. Then factor 5 promotes the dissociation of several other factors and responsible for association of 40S and 60S subunit to form ATS initiation complex. These are the different steps in initiation process. This is the animation showing the initiation process. These are the three sites which are present on ribosomal unit. The first step is ribosomal dissociation, then attachment of the factor and stabilize the 40S subunit, prevent the reassociation of these two units. Then there is attachment of ternary complex to form 43S pre-initiation complex. Then attachment of active mRNA molecule. This help in the formation of 48S initiation complex. Then there is recognition of AUG with the first codon. Then after attachment of 5B factor, eukaryotic initiation factor 5, which brings larger unit to form ATS initiation complex. All other factor dissociate after attachment of the eukaryotic initiation factor 5. Then elongation, it is described in these three steps. This is the animation of elongation. Incoming tRNA with amino acid get attached to the acceptor site with the help of factor eukaryotic elongation factor 1 and GTP. This diamond shape is the eukaryotic elongation factor 1 and black colored circle indicate GTP. Then peptide bond formation occur and chain elongation occurs. Then deacylated tRNA will be exit from exit site. Then there is attachment of eukaryotic elongation factor 2 along with the GTP, which is responsible for translocation of ribosome to the 1 codon. Now acceptor site will be free. In termination, there is a release of newly synthesized protein, mRNA, tRNA and ribosomal units. Release factor 1 and release factor 3. That orange color triangle represent the release factor 1, while blue color represent the release factor 3. Along with the energy, it brings about the hydrolysis of that peptide chain and peptide will be released. And all other factor dissociates one by one. This is difference between prokaryotic and eukaryotic translation. In prokaryote, translation is coupled with transcription and the mRNA in prokaryote is polycystonic. That means one mRNA code for multiple proteins. Initiator amino acid is in eukaryote is methionine, while in prokaryote it is formylmethionine. There are many factors required for eukaryotic translation. Ribosomal unit is 80S in eukaryote while 70S in prokaryote. In eukaryote, mRNA binding to the small subunit involves the cap at 5' end of mRNA and initiation factor, while in prokaryote it involves Shine-Dalgarno sequence which is present upstream of the AUG. These are the eukaryotic ribosome and prokaryotic ribosomes. In elongation, there is requirement of elongation factor TU and elongation factor G in prokaryotes, while 1 and 2 in eukaryote, and 1 and 3 required in eukaryotic termination, while 1, 2, 3 required in prokaryotic termination. Regulation is with the help of phosphorylation of eukaryotic initiation factor 2 and eukaryotic initiation factor 4E that regulate the translation. These are the inhibitor of protein synthesis. Prokaryotes, eukaryotes and both puromycin inhibit both translation in prokaryote and eukaryote. Is the tetracycline, chloramphenicol, erythromycin, aminoglycoside. There is MCQ on diphtheria toxin. It catalyzes the ADP ribosylation of eukaryotic elongation factor 2. And there is MCQ on puromycin, causes inhibition of translation in both prokaryote and eukaryote. This is all about eukaryotic translation process. If you like the video, please press the like and subscribe to the channel. Thank you, all of you.